Hello everyone, my name is Dr. Ambud Misra, and in this video we are going to discuss about fatty acids, its classification, and the process of lipolysis. So what is fatty acid? Fatty acid is basically your carboxylic acid, that is your a methyl group that is attached to a functional group, that is your carboxylic acid. So this methyl group is a variable in different types of fatty acids. And what is lipid? So lipids can be categorized into two types, that is simple and compound. So the constituents of a simple lipid is your fatty acid plus glycerol. It is made up of two subunits, that is your fatty acid and another one is your glycerol. And the example of a simple lipid is fat, oil, waxes, and your triacyl glycerol, that is your that is also known as triglycerides. So triacyl glycerol is also your simple lipid. What is your compound lipid? In compound lipid, along with your fatty acid and glycerol, you will see another component. You will see any other compound that will be attached to fatty acid and glycerol, and that could be different in different kind of compound lipid. Some examples are your phospholipid, or your glycolipid, and your lipoproteins. So in case of phospholipid, the other compound is your phosphate group. And in case of a glycolipid, it will be a carbohydrate group. And lipoproteins, when the protein is attached to any lipid, it is known as lipoproteins. So the lipids are transported into the blood by they don't exist in free form, so you are always going to find them attached to a protein group. So they are going to form lipoproteins. That is an example of your compound lipid. Now coming on to fatty acid. So fatty acid, uh, fatty acid is stored inside the adipocyte, in the form of triacyl glycerol. So it has two components. It is a form of, it is a simple lipid. It has two components. One is fatty acid and another one is your glycerol. So fatty acid is mainly stored in adipocyte in the form of triacyl glycerol. So triacyl glycerol is, as you know, this is made up of two components, the fatty acid and glycerol, where one glycerol is attached to three fatty acids. So this right here is your component of glycerol, and these are three different fatty acids. So this is a general formula or a general representation of a fatty acid, you can say. Now moving on to triacyl glycerols. So triacyl glycerol is, it comes from two sources, you can say. So it can come from intestinal absorption, or you can say intestinal dietary absorption. And another source is your liver. So through the process of lipogenesis, where fatty acid and glycerol combine in order to form triacyl glycerol. So via the process of lipogenesis, triacyl glycerols are formed inside the liver. So some points about triacyl glycerols. So these are the most abundant lipid inside the human body that comprises around 85 to 90% of all the lipids are in the form of triacylglycerol. It is stored in adipose tissues, or you can say adipocytes. So when one compound of triacylglycerol is broken down, it is going to release some amount of energy. So if you compare it from the proteins and carbohydrates, so triacylglycerols or fatty acids, when they are broken down, they are going to release most amount of energy as compared to protein and your carbohydrate. That is, it releases a significant 9 calories per gram of when 1 gram of triacylglycerol is being broken down, and it is stored in anhydrous form. It is stored in anhydrous form. So what is the significance of this being stored in anhydrous form is, why it is the best form of energy that body can use, best form of stored energy, that is your lipid, your triacylglycerol or fatty acid, because it is stored in anhydrous form and it is not going to attract the water. Whereas if you see carbohydrates like glycogen and other variants, they are going to attract water. So wherever they are going to exist, they are going to attract water. They will ultimately increase the amount, the body weight. So that means in order to generate the same amount of energy, you need more glycogen, whereas you need very less fat or triacylglycerol or fatty acid to generate the same extent of energy. So that is the significance of triacylglycerols being stored in anhydrous form. Now moving on to the process of lipolysis. So lipolysis starts when any hormones are going to act on the when in certain situations, when certain hormones are going to get released, which are going to further act on adipocytes, and on the surface of adipocytes, on the membranes, you have G protein coupled receptors. Okay, so these hormones are going to act on these G protein coupled receptors, which are present on the surface of adipocytes, on the adipocyte membrane, which in turn is going to stimulate adenylyl cyclase. This adenylyl cyclase in turn is going to convert ATP into cyclic AMP, and this cyclic AMP in turn activates protein kinase A, and protein kinase A further is going to activate one enzyme, that is your ATGL. What is ATGL? It stands for adipocyte triglyceride lipase, which will break down one molecule of triacylglycerol into diacylglycerol and one free fatty acid. Further, this diacylglycerol is another hormone is going to act on this, that is your HSL, that is your hormone sensitive lipase. Hormone sensitive lipase. This hormone sensitive lipase is going to break down a molecule of diacylglycerol into monoacylglycerol, and it will further release one molecule of fatty acid. So this is your fatty acid 1, this is your fatty acid 2, and this is your fatty acid 3. And another hormone, that is your MGL, which stands for monoacyl glycerol lipase, which is going to break down monoacyl glycerol into one molecule of glycerol and fatty acid. So this glycerol and these three fatty acids that we are going to obtain from one molecule of triacyl glycerol. Now, you saw that in certain conditions, like when you are fasting, when you are exercising, or in case when you are starving, or in case when your body has just taken food. So in all these conditions, the specific types of hormones are going to get released, which in turn is going to stimulate the process of lipolysis. So what are the hormones that are going to increase the process of lipolysis? We are going to see. So the hormones that are going to increase the process of lipolysis is your epinephrine, which is the most important, or even say most potent of all. You will also see norepinephrine, your glucagon, your thyroxine, and ECTH. So these hormones, when they are going to get released and when they are being acted upon the adipocyte, they are going to stimulate the process of lipolysis. And which hormone is going to decrease or suppress the process of lipolysis is your insulin. Insulin is going to suppress the process of lipolysis. Why? Because lipolysis is done when in certain conditions, like when there is deficiency of glucose inside the body, and body needs energy, but the glucose is not available for that. So body need some another way to comprehend that, and obtain the meet the energy demands of the body in when there is a deficit of glucose. So insulin, which is going to decrease the lipolysis, because insulin is released when there is glucose inside the body, like the in postprandial state, when you take food, insulin is going to get released only then. So that means glucose is already present, so the process of lipolysis is going to get suppressed in when insulin is being released. So what, by which mean these hormones are going to increase the process of lipolysis? They are going to do so by increasing the activity of adenylyl cyclase. They are going to do so by increasing the activity of adenylyl cyclase. And we saw when the adenylyl cyclase is going to get activated, it is ultimately going to facilitate the process of lipolysis. So the hormones which are going to increase the process of lipolysis, they act by increasing the activity of adenylyl cyclase. And how insulin is going to suppress the process of lipolysis? It is going to do so by decreasing the cyclic AMP. So when insulin is going to released, it is going to decrease the cyclic AMP. Here it is, and which further is going to suppress the process of lipolysis. So this is the process of lipolysis that we just saw. Now we have two components, the fatty acid and glycerol. So there is, what is the fate of glycerol in the process of lipolysis? So glycerol, it is going to transported to the liver. Okay, and in liver, the glycerol is being acted upon by this enzyme known as glycerol kinase. Glycerol kinase, which in turn is going to convert glycerol into glycerol 3-phosphate. Now what is going to happen with glycerol 3-phosphate? There are two ways that glycerol 3-phosphate is going to get utilized. First one is your synthesis of triacyl glycerols and your phospholipids. And the another one is, it is going to enter the process of glycolysis. It can directly go and enter into the process of glycolysis, which will going to form pyruvate, and pyruvate further when it is going to get acted by the enzyme pyruvate dehydrogenase, which is further going to form acetyl coenzyme A, which is going to enter a TCA cycle or Krebs cycle, and which is further going to generate energy. Or it can again go back for the synthesis of triacylglycerols and phospholipid, that is your compound lipid. So this is the fate of glycerol. Now we will see the fate of free fatty acid. So free fatty acid, fatty acid cannot stay inside the body or inside the blood circulation alone. It need to be bound to some another compound. So if free fatty acid is in circulation, it is bound to albumin. So when the process of lipolysis occurs inside the adipocytes, the glycerol and free fatty acid are going to get released outside, from the adipocytes into the circulation. So glycerol is going to reach liver from the circulation, and free fatty acid, which is released in circulation, first it is get bound to albumin, and when it enters the tissue, so in cells it is bound to fatty acid binding protein. In cells it is bound to fatty acid binding protein, and whenever it reaches the cells, then there occurs the beta oxidation of these fatty acid. So beta oxidation, in which types of cell the beta oxidation occurs? It occurs in all types of cells except brain and erythrocytes. Except brain and erythrocytes. And further, the process of beta oxidation is going to lead to formation of energy. Now, you can see in the process of lipolysis, we saw like fatty acid and glycerols are going to get released, but 65% of this fatty acid is going to re-esterified into forming triacylglycerol again, and this triacylglycerol is going to get sent back again to the adipocytes. So it is said that 65% of this fatty acid is re-esterified to triacylglycerol and sent back to adipose tissues. Sent back to adipose tissue. Now, we saw the fate of glycerol and we saw the fate of free fatty acid. Now, we can classify the fatty acid based on some properties. So the first classification that we need to learn is your omega classification. So based on the position of first double bond from the terminal methyl group, not from the functional group. Like we generally, when we do the nomenclature of chemical compounds, we start counting from the functional group end. But in this case, in the case of omega classification of fatty acid, the counting is going to get start from the methyl end, from the terminal methyl end. So let's just say this is your fatty acid, and from here, from this side, if we are going to start the counting, because this is your terminal methyl group. So from here, first, second, third, and fourth. This is your fourth carbon group where the double bond first appears. So this will be your omega 4 fatty acid. Okay. But there are two main types of fatty acid, omega fatty acid, that we need to learn. That is your omega 3 and omega 6. So omega 3 and omega 6, these are your essential fatty acids, and these must be obtained through your diet. These must be obtained through diet. Now we can see some examples, some main examples of omega 3. So alpha linolenic acid, alpha linolenic acid, and your timodonic acid. These two are omega 3 fatty acid. Remember, this is your linolenic, not linoleic. And omega 6, the example is your linoleic acid and arachidonic acid. This is your arachidonic acid. So these two main examples that we need to learn of omega 3 and omega 6 fatty acids. Alpha linolenic is omega 3 and linoleic is your omega 6. And timodonic, omega 3, and arachidonic is your omega 6. So we will see some importance of these fatty acids. So omega 3 fatty acid, it is going to decrease the cardiovascular risk, it is going to decrease the platelet aggregation, it is going to decrease the inflammation. And there is one type of omega 3 fatty acid that you must have heard about it, that is your DHA. DHA is very important for infant brain development. So DHA is a type of omega 3 fatty acid. So DHA is required for infant brain development. And we saw two examples of omega 6, that is linoleic and arachidonic acid. So this is the source of eicosanoids. What is eicosanoids? Prostaglandins and leukotrienes. This is your prostaglandin and this is your leukotrienes. So omega 6 fatty acid, especially your arachidonic acid, is precursor for the formation of prostaglandins and leukotrienes. These are your inflammatory mediators and whenever they are going to get formed or activated, they are going to cause inflammation, which in turn increases cardiovascular risk and increases risk of degenerative disorders. So arachidonic acid is an example of omega 6 fatty acid, which is going to form eicosanoids, which the examples of eicosanoids are prostaglandins and leukotrienes. These are inflammatory mediators which when they are going to get activated, they are going to increase the cardiovascular risk, and they are going to increase the risk of degenerative disorders. Moving on to next type of classification, that is based on double bond. It was also based on double bond, but it was based on location of the double bond, the omega classification. And the another classification where you can classify these fatty acid based on number of double bonds into saturated fatty acids and unsaturated fatty acids. So in saturated fatty acids, double bonds are absent, and in unsaturated fatty acids, double bonds are present. Basic. Now you see saturated fatty acids, we will see some examples of saturated fatty acids. So saturated means no double bond, that we already know, like no double bond. Some examples are that we need to remember. The first one is your palmitic acid. Palmitic acid. Palmitic acid, what is the source of palmitic acid? It is your butter, your cheese, and your palm oil. The second one is your stearic acid. And the sources are your animal fat and your cocoa butter. The third one is your lauric acid. And the source is your coconut oil. And the fourth one is your acetic acid. And we have learned this in school that the source is your vinegar. Okay, now these two are your long chain fatty acids. This is your medium chain fatty acid, and this is your short chain fatty acid. Based on a number of carbon atoms, we can also classify this into short chain, medium chain, long chain, and very long chain. So this is not really that important, and no one is going to ask you. But for the sake of knowledge, these two are long chain fatty acids, that is your palmitic and stearic. Lauric is your medium chain fatty acid, and acetic acid is your short chain fatty acid. And the sources that I already told you. Now, unsaturated fatty acids can be categorized into two types again, that based on a number of double bonds. So, MUFA and PUFA. MUFA means monounsaturated fatty acid, and PUFA means polyunsaturated fatty acids. So, in MUFA, there is single double bond, and in PUFA, there is more than one double bond. And the examples are your, palmitoleic acid, the examples of your MUFA. And the source is your mustard oil. And another one is your oleic acid. The source is again mustard oil. Nothing very important, nothing very specific. Now moving on to PUFA, polyunsaturated fatty acid, which contains more than one double bond. The first example is your linoleic acid, which we saw here, which is your omega-6 fatty acid. It is also an example of PUFA, that is your polyunsaturated fatty acid. And the source is your safflower oil. Not sunflower, it's safflower. Then again, another one is your gamma-linolenic acid, and the source is your oil of evening primrose. Then alpha-linolenic acid, and the source is your flaxseed oil. And then comes your arachidonic acid. So arachidonic acid, it is the substrate, we saw like it is the substrate for synthesis of intracellular regulators, that is your prostaglandins, thromboxanes, and your leukotrienes and all these. So this is the substrate for synthesis of intercellular regulators, that is your prostaglandins, thromboxanes, prostacyclins, and your leukotrienes. Next is your cervonic acid, that is your DHA, and it is the full form is your decosa hexanoic acid, and it is found in fish oil, and breast milk. The source of DHA is fish oil and breast milk, and it is responsible for visual acuity, and brain growth of the newborn. So that is, DHA is really important for newborn, or you can say baby. Now moving on to another classification, that is your you can also classify fatty acid into cis and trans fatty acid, that is based on your geometric isomerism, where you already know, if the same type of functional groups are on the same side, that is your cis. If they are on opposite side, that is your trans. So if we we can further see cis, the significance of these. So the cis fatty acid, these are going to increase the fluidity of plasma membrane. So cis fatty acids are important for increasing the fluidity of plasma membrane, and the trans, it has multiple functions that we will see. These are, you have seen, you have learned from many, uh, you must have learned this from your early days, that trans fatty acids are really harmful. And so what makes it harmful actually? So these are partially hydrogenated fats. These are partially hydrogenated. These are harmful, and they are formed by deep frying or reheating the vegetable oil. So for example, if you are refrying some kind of food, or if you are reheating any kind of vegetable oil, if you are using it for the second time or third time, so the fatty acid is going to convert from cis to trans, and which is really harmful. So this is formed by you know by deep frying and reheating of vegetable oil.